we realize we're not giving you an official break, but if people need to leave to get a cup of coffee or something, please feel free to. We just want to make sure we give all our speakers enough time, and we're running right on time right now, but we don't want to fall behind. So our next speaker is Professor Andrea Malaspina from Queen's Mary's College, London.
I would like to thank, first of all, the organizers for inviting me to this conference. I'm really glad to be in Amsterdam, which is a city I love particularly. So essentially, I would like to drag you onto a less promising and delightful theme, which is divided of all the therapeutic successes which we've heard about in MS. In the field of neurodegeneration, essentially, as Kevin has shown this morning, there is a retreat from investment in terms of new treatment for what I can see, with some timid, optimistic developments in mostly monogenic disorder like SMA, which is a neuromuscular disorder. One of the main effort is also to try and develop a concept of preventative neurology. So essentially using our clinical observation and biomarkers to identify individuals that are at risk and go into a safe zone in which we can start treating or counseling. And this is taken from Alistair Noy's exceptional work where I live and work in London, and you can see how in Parkinson's disease, for example, you can have some element of clinical observation that can hint to a risk of later development of the disease. And this is obviously correlated with the potential of early neuronal loss leading to the disease. So I'm not going to bother you with what a biomarker should be in terms of early diagnosis, in terms of understanding and dissecting the speed of progression, and above all, giving you an idea about response to treatment. So I was in Glasgow last week at the World MND Symposia, and there was a debate. I was asked to participate on what is the attitude that drives essentially the research interest and eventually also the development of biomarkers and therapeutics. So are you a splitter or a lumper? Do you tend to take a disease as an entity and therefore you develop treatments that are comprehensive and effective? Or are you down to details and you want to spend more time in stratifying and define the right phenotypic variant and the treatment that go along with it? And clearly, if you are in a muffin tasting competition, I think you have to pay attention not to eat the chihuahua. Amyotrophic lateral sclerosis is a neuromuscular disorder. It's probably what you were missing today, and it's a completely different paradigm of neurodegeneration. You can see that the diagnostic latency, for example, ranges still in the Western world between 12 to 15 months. Despite the fact that clinical tools, so somebody like me who has more than 20 years neurological practice and a good eye for what is ALS, are good and functioning. And the other important aspect of this disease, which I think characterizes the entity against what we see normally in neurodegeneration, is the profound clinical heterogeneity. So in terms of speed of disease progression, which is essentially what we work on, you can come across a patient that may die in 6 months from your first appointment, and at the same time, the same clinical manifestation in terms of upper and lower motor neuron involvement would make somebody survive up to 2 decades. there is essentially an interplay biologically in terms of disease progression of different level of involvement that go from the neuromuscular junction to the cortex. Most of these individuals develop sooner or later some form of frontotemporal dementia. There is an interplay with autoimmunity, profound derangement of energy metabolism, and as we've heard in the last few years with mechanical injury or repetitive trauma. What you see here is essentially what I've just mentioned, is the fact that you have a number of sites where the disease may be considered initiating, and the whole spectrum of this anatomical location are essentially where the disease can then propagate. And on the right side, you see the very rich genetic background that this disease entails, and a number of genetic mutations have been discovered in the last 15 years and encompass about 10% of the individuals and about 70% of the familiar form of the disease. This is to give you an idea of one of the things that is attracting more attention at the moment in terms of these neuromuscular conditions. I've plotted around the previous figure all the inflammatory mediators that have been found to be modified as a consequence of the progression of the disease. And this is both a humoral type of response and an innate type of response. And this is both at systemic level, so in circulation, and within affected tissue in autoptic studies. So this is what we have created over the years. I think it's an interesting experiment. You have to forgive me, I'll put a few pictures in the following slides because I think it's important. You see the faces of people driving the main efforts in this. So we have constructed a biological repository and of clinical data which encompasses disease events, so the natural history of the disease, including the various phenotypic variants, so the fast versus slow progressing individuals and the limb versus bulbar onset individuals. And we have essentially teamed up with different individuals in the US. This is Mike Bennett at the University of Miami. This is Len Petrucelli at the Mayo Clinic. These are other collaborators in the UK. And essentially put a tail which is represented by the longitudinal serial sampling and clinical data collection from individuals at risk of developing the disease, who are essentially mutation carriers. So this has been an enormous undertaking, but this means that we have samples of different types that stretch back up to 2 years before the disease onset, and therefore we can reconstruct biomarkers-wise the development in a silent clinical state and then in a prodromal phenoconversion phase of the disease. And then obviously go into what the disease is in terms of clinical progression. So we managed to get funding to sustain this, and we have somehow pooled all the resources together. It is difficult to collaborate. I agree with somebody who raised this issue. One of the things we've done is we have invested on a liquid biopsy type of concepts. I am a proposer of blood as a main matrix for developing biomarkers, although it's very important for CSF being included, is the repository of neurosecreted or products of destruction from neuronal cell. The blood gives you sometimes a better angle of observation in terms of immunomonitoring. It's certainly important in the concept of microsampling. There is a presentation later on, I just heard. And we have, for example, dry blood spotted and dry plasma spotted. All the individuals have been biobanked since 2009. And in parallel now developed some sort of confirmation that we can get neurofilament light chain, for example, reading from elute from dry plasma and dry blood spot. So this is one of the examples. This is not about only neurofilaments. So I'll give you a bit of insight about what we've done on established biomarkers and also on discovery effort. This is Mary Louise Rogers from Australia, who has been quite good in developing a urine biomarker, which is actually quite interesting in the ALS world. One of the things that characterizes it is the fact that it has an upward trend in the progression of the disease, unlike neurofilament light or heavy chain. And we have some preliminary data on a clinical trial, which I'm not going or able to mention, but essentially we have a very good treatment response based on the urine biomarker. Urine is obviously a very complex matrix. It has to be normalized in terms of measurement with different parameters. And obviously we have done our bit on neurofilament light chain. This is a study we've done in collaboration with the University of Oxford, in which we used 2 longitudinal cohorts and addressed the question whether neurofilament would give us a good stratification in fast versus intermediate versus slow progressing individuals. And this work was only possible thanks to the monumental effort of Jens, who was undertaking also some doctoral work there, and we have managed to pull out some interesting data on how to use neurofilament light chain with a prognostic significance, being able to understand a bit the progression of the disease based on a baseline measurement. We've also managed to use neurofilament light chain and heavy chain to test the longitudinal samples we have collected, and what you can see here on the left is essentially a fairly flat line neurofilament levels for all subgroup of patients with ALS over a period of observation of about 24 months. For neurofilament heavy chain, we can observe a steep decrease of levels in fast progressing individuals. And essentially what we've done, we have replicated the same type of observation using animal model of ALS, which are engineered with a transgenic mutation, a human mutation in a specific genetic strain, and obviously demonstrated the sparing effect of one of the medication now on trial for ALS is arimoclomol has in neurofilament levels, the ability to reduce the area under the curve in the lifespan of a mouse. And this is the work we have done originally with Mike Benatar, who has collected all the pre-symptomatic familial ALS individuals. And you can see here on the left side levels of neurofilament light chain in individuals who are carrier of different genetic mutation linked to ALS compared to control. And you can see when these individuals develop the disease, the neurofilament level tends to increase quite significantly. And here you see the longitudinal time point. These are only a few samples, a few patients. The cohort of converters has now increased. We have many more individuals who have developed the disease. And what is interesting is that at time 0, patients are seen in clinic and they present some features, normally very soft, of upper and lower motor neuron involvement. Also, they are electrophysiologically indicating that the disease is present, but they're completely asymptomatic. So this data show you how we can think of a prodromal period in which the disease is clinically silent, but neurofilaments start increasing quite significantly. And clearly, if you have a treatment, you will be able to essentially treat timely in this moment of time. This is another neuromuscular disorder similar to ALS, but is much milder. It's genetically inherited. It is characterized by an expansion of the androgen receptor and has a clinical feature mostly of lower motor neuron involvement. For those who are probably not familiar, this means a kind of flaccid weakness with a lot of muscle activity. And it has been traditionally considered a lower motor neuron disorder. So the onset and propagation of the disease, presumably in a neurological structure. But essentially, we've seen comparing spinal bulbar muscular atrophy from the UK and Italy in different cohorts to slow and fast progressing ALS individuals that there is no neurofilament signal here. They have no signs of these biomarkers increasing in blood. Whereas if you use biomarkers which are essentially readily assessed also through NHS or hospital technology like creatinine and creatine phosphokinase, you see that they are much more representative of the disease compared to ALS. And they actually correlate very well with the clinical scores that we are normally using for the monitoring of these motor cell disorders, which are mostly related to functional disability, in this case of lower limbs. So is spinal muscular bulbar atrophy a disease of the muscle? This is counterintuitive clinically, but that's probably what comes out also from animal studies and certainly also from this investigation. What we've done is also to use both imaging and neurochemical markers, including neurofilaments, in the context of a multimodal approach. This is a small study we've done in which we looked at fractional anisotropy in a DTI MRI scan and neurofilament. We find very good correlation between CSF neurofilaments and the FA parameter. This is to give you an example of the fact that ALS has got some form of MRI positive findings in the corticospinal thread. Now on the right side we've done the same thing. So we use imaging and clinical outcome measures and biomarkers in SBMA and shown some specific pattern of involvement and progression in muscles of the lower limbs of the posterior compartment of the leg and also in the bulbar region, differentiating, I think, quite neatly ALS from SBMA. But another important aspect of this neuromuscular disorder is the inflammatory response. I just briefly hinted at it before. And one of the main findings that has moved the field of immunotherapy of ALS, which is still in its embryonal stage, along the lines is the fact that individuals with ALS seems to have a profound downregulation of T regulatory cells in the disease, and it is one of the best biomarker of disease progression. So the faster the disease, the lower T regulatory cells we find. So this is simply a FOXP3 and CD25 study, the original study, which has been replicated, and I'm fully aware that T regulatory cells is a complete, is a very immunologically dense topic where there are a lot of subtypes that may be implicated in the disease. And so, but this led to one of the largest clinical trials in ALS so far, sponsored by the European Union, of which I'm part, and I'm the biomarkers bucket lead. And it's essentially using IL-2 to try and push T regulatory cells up with the aim of overcoming this important immunological change. The trial is ongoing, is one of the few ongoing in ALS, and the jury is out whether there is any significant effect. If you take a simple platform and you measure systemically cytokine expression and also acute phase reactant, you can still have a good signal which separates your ALS patient from your healthy control. Obviously, these are very well-defined cohort of individuals without any systemic autoimmune disorders. They are still chosen based on the absence of any major element of medical history, and we do some multivariate analysis. And you can use IL-6 to somehow look at the progression of the disease, and you see certainly an increase over time. One of the things we are doing at the moment is also looking at microRNA and cell-free DNA in the progression of the disease in collaboration with the Weizmann Institute. And we have very interesting data which correlate quite well with plasma NFL levels and TNF-alpha, which are also two biomarkers we're using in this context. So data are being analyzed as we speak and are shown here. Neurofilaments are essentially interesting. I'm not an expert in neurofilaments, but what we have been always interested in is the behavior of neurofilaments in biological fluid. So if you do, for example, a dilution curve and use your standard protein, this is NFH, you have linearity. Whereas if you do the same thing on your plasma samples, you have what we describe as a hook effect. So essentially what we think is happening is that neurofilaments may become sequestered by in immunocomplexes or get into protein aggregates in the peripheral circulation. You can see here, if you stain for your neurofilaments, you have the band, the expected molecular weight, and other high molecular weight bands. So somehow stumbling onto this observation, we have become interested in the concept of immune response or humoral response to the release of neurofilaments. So we have somehow started testing antibodies against neurofilaments for the purpose of stratifying the disease. And you can see here that you can somehow see an increased level of neurofilaments as the disease progresses, so in an early, intermediate, and advanced stage. We've also worked on our samples to pull out these neurofilament containing aggregates and trying to understand whether they have any resemblance with the insoluble fraction from brain, so whether they carry some of the information in terms of composition of aggregation within brain. Talking about antibodies against axonal protein, we are in collaboration with industry, with partners that have technology to develop antibody libraries, and somehow what we're interested in is in developing antibody libraries from phenotypic streams of the disease. So individuals that, for example, develop the disease in a very fast and aggressive way. So we essentially use our biological samples in a phage display library approach to characterize the antibody library and specific antibodies against antigens we are interested in, including neurofilaments, hoping to get targets that may be used as essentially a reagent for screening a particular disease phenotype, or perhaps for stratifying patients for clinical trials. The very last thing I wanted to mention that is part of our discovery effort is essentially a new technique of proteomics which we have applied to the paradigm of fast versus slow disease progression. So we are used to the fact that omics are normally quite informative as long as you test your RNA, your DNA. But when it comes to proteomics, you're always very limited when you're testing your biological fluids, particularly plasma, by the fact that you have a low abundance protein which are masked by your more abundant proteins like albumin and antibodies. So we have partnered with a company has developed a technique that allows you to look at tissue and fluid in the same templex. And your tissue will be essentially your internal calibrator, so your standard curve, and you will be using TMT labeling for your mix of tissue and fluid. So what comes out in our comparative study of fast versus slow progressing individuals is essentially a profile of protein which are expressed in the brain, but are also detected in plasma with their differential regulation. And among these low abundance proteins, we have essentially detected proteins that wouldn't normally be detected using MS proteomics, like, for example, SOD1, TDP43, and neurofilaments themselves. And so what we've done, we used both brain PBMCs, which are a readout of systemic inflammation, and we use also our circulating protein aggregates and brain aggregates, and look at the essentially composition which bridges tissues and fluid, and got a very good idea what molecular features or pathways are linked to speed of disease progression, and a number of interesting low abundance protein candidates, which we have now obviously to put through an orthogonal technique of validation, which could be SIMOA or the best available technique. So essentially this is what we've been doing, and we're doing in the field of ALS, which I believe is a bit of a minor world in comparison to the advances we see in MS, but we're trying to learn from the MS field to calibrate our effort in the right direction. Also because as a clinician with, as I said, more than 20 years experience in neurology, I find more and more fatigue in coming out of a clinic after having seen the devastating reality of this disease. The highest number of individuals asking for assisted suicide or undergoing traumatic suicide, and unfortunately families left in tatters when the disease ends. This is a bit the whole lot of people have been working on, have been working with throughout the last few years, and have been contributing largely to what I've just presented. Thank you very much.
Any questions?
Thank you very much, and I understand I share with you the concerns about ALS and the finding a target one day. One day we'll find it, but it's not there yet. My question is, or maybe the difference with MS, there were effective treatments, and then we apply the biomarkers on that. The world is very different. But from ALS, where do you see the most promising target where the disease could be improved, and you could then obviously measure better the biomarkers? What is your take on that? Because there was many drugs tested in ALS in the past, and many efforts done, but is there nothing really promising? Well, but where you see the field moving from a treatment perspective?
That's a very good question. There's a lot of discussion about also what is ethical or not ethical in terms of treatments for ALS. For example, patients with ALS don't want to be on a placebo arm. I guess it's the same thing with pancreatic cancer, they don't want you to stick a needle in their back. They tell you to go away after they become seriously ill. So my view of ALS is that it's really a condition which is enriched with a lot of genetics. Essentially, we're left with probably rare variants, but most of the big genes are there. But at the same time, people go and look at the pathology or systemic effect of the disease and look at the same things which are across different neurodegenerative disorder. So what is interesting for us is to note that for example, in ALS you have a phenomenon of aggregation both centrally and peripherally. The circulating protein aggregate is very similar to what you have in brain, and we managed to get it out of this proteomic technique, in which you have a real convergence of proteins that have a tendency to aggregate in different conditions, different degenerative disorders. So what people are doing at the moment we are involved in this is to go back from the actual biological information of the signal to the genetics, and trying to understand whether we can find a polygenic signature of the disease, which could be diagnostic. But at the same time, work on the protein aggregation aspect. What is coming out also quite quite interestingly, also from our work, is that there is clearly a very dense immunological response, a humoral response. This protein aggregates I've shown you, which we image with electron microscopy, immunogolden electron microscopy, are big immunocomplexes. So we started looking at neurofilament, for example, as disordered protein, oxidized, and trying to develop both antibodies that can target them. We have actually interesting results. I don't think that immunotherapy would work in ALS. I think it's an event that is somehow not primarily immunological, but has a strong immunological component that's driving the disease. But one of the things that is coming out quite interestingly, and we have very interesting data from our clinical trial in Australia, and we are now sort of developing the trial in Europe and UK, is the antiretroviral activation. So the HER-K story, which is now also coming out in other neurodegenerative disorder. And this is from a paper from Avindranet showing that motor neurons in animal model and pathological tissue are really enriched with this, really an example of reactivation of this. And quite interestingly, an initial trial shown interesting results, and also biomarker response, which is also related to the activity of transcriptase and reverse transcriptase, and also showing a P75 in the urine response. I have no other ideas.
Great. Thank you so much, Professor Malaspina.